An incident with the police.
Hello everyone. Let me check if I'm live here. Just a second. All right. So let's get started. I hope I am audible and visible. Alright, so let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Alright, so join with us in Telegram. So that is for homework and class notes. You can join the channel Vani Kari ICSE class 9 and 10 biology and the Telegram channel of Unacademy ICSE and ISE both. So this link you can find in the description box. So go to the description box. So there you will find the link of Unacademy ICSE and ISE both. You will find the link for the Telegram channel of this and also the community, and there is one more link. Yeah, that is Bugs Bounty link you will find, and also you can find some of these special classes link. Okay, so you can go to this description, click on the specific link to join in there, and if you have to any question, so let's say you have any doubt, so you can clear your doubt from the community or from the Telegram channel itself. Okay, so we conduct special classes on the platform, so that is Unacademy platform, where you can learn from different educators for free, because these are free classes conducted on the platform. Okay, all you have to do is download the Unacademy Learners app and start taking up the sessions. Once you are comfortable, maybe you can go ahead with a plus subscription, because in special classes only certain percentage of topics will be covered, whereas in case of plus classes, so there is 100% syllabus coverage that is done. So you can learn from your favorite educator for free. You can also engage with the educators via poll. So here we have come up with different theme based special classes. So different educators are having their own theme based sessions. So you will have access to their quizzes. So some of them are conducting quizzes, there is a topic teaching that is going on, so you will have access to their quizzes, because on the platform itself we can conduct a quiz. All right, and here with that, basically you can choose to participate in that. So once the quiz is done, so basically you can have a look at your leaderboard, so that you will know your rank and compete with your friends as well. Okay, so clear your doubt in the live special class. So there is a separate question tab where you can click on that. Basically you have an option to type in your question, attach an image and send it to the educator during the live session itself. So they'll be getting a notification that the learner has asked the doubt and will be clearing your doubt. Okay, so now for the top special classes, you can visit the link in the description. I hope I am audible. Okay, so Unacademy subscription features, so these are called as plus classes. Okay, so you can take the plus classes with the subscription there, where the features are you can learn live from the comfort of your home, unlimited access to all the courses. Now different educators will come up with different courses, so you will have access to their courses once you take the subscription. Okay, then top educators of India are going to take the classes in one platform, so that is Unacademy platform. A regular doubt clearing session will be done, so that is after about every 3 sessions. The 4th session will be our doubt clearing session. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in the doubt clearing session. All right, then so there is a exhaustive coverage of syllabus mentioned. So that is basically Here in plus classes we'll be covering 100% syllabus coverage will be done. Okay, so in special classes only certain percentage of topics will be covered. So mentorship and guidance will be given, study material will be given, and also practice tests are conducted, live test series are conducted, and also you will have access to batches. So we have a batch in English, we have a batch in English. So you will have access to both the batches once you take the subscription. And then there is daily practice section, weekly mock test series are conducted, live quizzes, daily MCQ and subjective test series are also conducted. So get up to 20%. So there is an offer going on right now. So that is unlock 20. All right, and you can use the code Vani YT. So this is my code. So with this code basically you can get 20% discount on the plus subscription, and this offer regularly it will be just 10% discount will be there, but right now from the July 17th to July 22nd, so we have this offer where you can get 20% discount on the subscription by using the code Vani YT. So don't forget to mention the code. So if you are going for 9 months of subscriptions, it is basically 14,615 instead of 17,000, and so basically you will be saving up to 2,578. All right, and here also now you can see this. So 19,838 is given, so which is now the offer price is 16,863. Okay, and again you will be saving up to 2,975. So if you are going for 18 months of subscription, so we always usually recommend you all to go with the 12 months of subscription, because once you get the 12 months of subscription, so you will have free access to class 10, class 11 and class 9. So since I'm taking for class 9 right now, okay, so class 10, class 11 and class 12 courses also will be free for you if you are going for 12 months or more than 12 months of subscription. Okay, so in that case it is 18 months, 24 months is given. So with that respect, so you can have a look at these numbers, and there is an offer price which is there, and you will be saving this much amount. Okay, so don't forget to mention the code there, that is Vani YT, to get 20% discount on the subscription. So these are the regular prices which are there, so you can go ahead with this one. All right, so today we are going to discuss about the protoplasm. Yes, has anybody has any doubt? Let me just check this. All right. Okay, so today we are going to discuss about the protoplasm. So what exactly is protoplasm? Any idea? When do we say that this is the protoplasm? So what cell can we directly mention this as the protoplasm? So since we are discussing about protoplasm today, any idea on this? What exactly this is? Is it a cytoplasm? So protoplasm, when we mention, so protoplasm is basically, it is the cytoplasm. Okay, so there is a cytoplasm, and this cytoplasm along with the nucleus. So cytoplasm and nucleus, which is covered by the cell membrane. So covered by what? Covered by the plasma membrane, or which we can also call as the cell membrane, right? So this we call as the protoplasm, right? So there is a cytoplasm. So what exactly is the difference between this? So what exactly is the cytoplasm? So cytoplasm, when we mention cytoplasm, is a semi-fluid, okay? So this one is semi-fluid, And this semi-fluid consists of what? So this consists of many different organelles. So you can find different organelles in there. Okay. And now nucleus is what? So nucleus is basically there is a DNA. Okay. So that is the genetic material will be there. So, and this genetic material, so we can just mention the DNA or the RNA also. So it can be any genetic material. So that will be present inside. Okay. So now whereas this whole thing, so there has to be covering. So that is covered by the plasma or the cell membrane. So that we call as the protoplasm, isn't it? Yes. Is it what we call as protoplasm? Or is there anything else which can be called as a protoplasm? So protoplasm, when we mention, so there is one more thing that I need to mention. So this is, okay, I'll explain you this just a second. So protoplasm, this can be considered under the living content of the cell, right? So this is the living content. So what all comes under the living content of the cell? So this is with respect to living content of cell, all right? And in that living content comes the cytoplasm, because cytoplasm, when we mention, so there are a lot of metabolic activities that are occurring. Okay, so this is with respect to cytoplasm. Just a second. Yeah. So now in cytoplasm, so you can see many different activities that are occurring. Okay, so what are those different activities? So many metabolic activities are occurring. Okay, so in this semi-fluid medium, so there is a semi-fluid medium which consists of organelle, all right, and the suspended proteins in the semi-fluid. So in that case, there is a metabolism that occurs. So there can be catabolic or anabolic reaction. So there is a catabolism, okay, and there is an anabolism which can be seen. So catabolic reaction, anabolic reactions can be seen here, right? So now what exactly is the catabolic? Why do we say that the cytoplasm is a living content, or on the whole the protoplasm? Okay, so protoplasm, we mentioned that it is a living content. So why exactly do we say that? Because there is a continuous metabolism that is occurring. So there can be constructive reaction. So this catabolism, anyway, it is a destructive reaction. Oh, just a second. Yeah, so now this part, what do you see? Okay, so this one is the destructive reaction. So there is a destruction occurring. So there is a destruction of what? So example we can mention, so there is a breakdown where the starch is basically converted into glucose there. Okay, so there is a breakdown process which is occurring. Okay, so starch is converted to glucose there. So many units of glucose are formed. Now anabolism is what? Anabolism is construction. So there is something that is being built up. Okay, so there it can be with respect to anything. So again, if we have to take, so many glucose units are joined together and there is the formation of starch. So what you can see here, here the construction is occurring and here the destruction is occurring, right? So catabolic reaction or the anabolic reaction, when we mention, so this is a destructive reaction and this one is the constructive reaction. So there are metabolic reactions that keep happening in the cytoplasm and also in the nucleus. So cytoplasm, since the cytoplasm contains many different organelles, so there are organelles involved in here. Okay, and with these different organelles that are there, okay, so there are different organelles. So with these organelles we can just mention that basically there are metabolic reactions occurring in a different organelles. Okay, so there can be modification of protein might be occurring. All right, so let's say modification of protein that is occurring there, or there can be synthesis of protein, synthesis we can say, so synthesis of carbohydrate, then the lipids. So then you can see also there might be formation of protein that is occurring. All right, so different functions are occurring at the cytoplasm itself. So there are different functions. It can be metabolic itself. Okay, so there are different functions which are occurring, which can be metabolic in nature. So that is occurring where? In the cytoplasm, including those organelles. Okay, and that's the reason the protoplasm, when we mention, so it is a living content. So protoplasm is a living content. So let's just discuss that. Okay, so but before that, let's have a look at this. So main features of a cell. So now this one, what you see here, so this itself is the protoplasm, right? So how can we say that this itself is a protoplasm? Any idea? Am I audible? Alright, so structure of a cell. So there are main features of a cell. So there is cell membrane, nucleus, cytoplasm. So all together it is a protoplasm itself. So now when we mention about the protoplasm here. Okay, so here I'll just draw. Okay, so protoplasm, when we mention, just a second. I think there is a network issue going on from my side. No, I guess. Okay. So now, with respect to this, we can mention protoplasm is nothing but, so there is a cell membrane. So this is now an animal cell. Okay. So animal cell can directly be called as the protoplasm. Okay. So this is the animal cell. All right. So an animal cell, it is a protoplasm itself, because it is not having that dead covering. So that is the cell wall. So cell wall, if it is there, so that has to be removed. So in this case, it is the cytoplasm inside here. Okay, so covered within the membrane. So this one anyway, it is a cell membrane. All right, and here you can find this nucleus having its genetic material. So animal cell is directly, it can be called as the protoplasm. But if we have to call a plant cell a protoplasm, so this extra covering, what is there? So that is a cell wall. So cell wall has to be removed. So I'll just explain it to you here itself. So now this is a plant cell. So there is a cell wall. Okay, so now we cannot call this plant cell directly as the protoplasm. So from plant cell, remove cell wall. Okay, so we need to remove cell wall, and then the remaining content can be called as the protoplasm there. So now this is the cell wall. Okay, and then there is a cell membrane that you can find inside this. So let's say there is a cell membrane which is present over here. All right, and this one is cell wall. So what is this? So this is cell wall. This one is what? This is cell membrane. Okay, and then you can find there is a large vacuole that you can find. So about 90% of the space in case of plant cells, so that is covered by the vacuole itself. So we can just draw the vacuole here. So there is a vacuole which is present. So this one we can just label this as the vacuole now. Okay, so which contains the cell sap. All right, then the surrounding material. So what exactly is there in the surrounding? So surrounding, it is basically a cytoplasm with different organelles. So about 90% inside the cell membrane. So that is basically covered by the vacuole itself, which is providing a turgid condition to the plant cell. Okay, so now when do we say that the protoplasm in case of a plant cell? So if we remove this cell wall, all right. So we need to remove the cell wall, then this can be a living content, and then because the cell wall is a dead content, so let's say this, I'll just erase this. So now we have removed this part, so that is cell wall is removed. So in that case, this can be called as the protoplasm after removal of the cell wall, or otherwise the plant cell on the whole in general cannot be considered as the protoplasm. Okay. So then other than that, so let's have a look at one of the examples, eukaryotic cell. So eukaryotic cell, so this one, let's talk about the animal cell itself. Okay. So we'll talk about the animal cell as an example. All right. Since I'm discussing about the protoplasm, so plant cell cannot be directly called as a protoplasm because it is having one more covering, so that is the cell wall, which has to be removed and then can be called as a protoplasm. For animal cell, directly we can mention that this is a protoplasm because there is a cell membrane, and you can find, so that is cytoplasm and a nucleus is present. So now the animal cell, so since it can be directly considered as the protoplasm, so we'll have a look at the features of the cell there. Okay, so that is with respect to animal cell. So in this case, so size, it is going to vary, that is between 5 micrometer to 100 micrometer. Okay, so nucleus is well defined, and that's the reason we call this as a eukaryotic cell, right? So it is U is what? U is true, so there is a true nucleus. So number of chromosomes, so they are anyway going to vary, but yes, there are more than one. The type of cell is an advanced type of cell. The number of cells, so there can be single cell or there can be many cells which can be present. So membrane bound organelles, yes, they are present, like Golgi body, endoplasmic reticulum. So then you can find a lysosome, then a vacuole. So these are membrane bound organelles that are present. So ribosome is also present, which is a larger, or it can even be attached or free there. Okay, so components of eukaryotic cell, when we mention, so this is for a plant cell. We are anyway not considering plant cell now, because since we are discussing about the protoplasm, so this cell wall has to be removed there, and rest of it is all protoplasm. So now the cell membrane is present, endomembrane system, so that includes endoplasmic reticulum, Golgi body. Okay, so then the lysosome, vacuoles can be included in this. So this is lysosome and a vacuole. So other than that, you can make a note here. So there is a mitochondria, there is plastid. Okay, so you can find a ribosome, then there is cytoskeleton, cilia and flagella can be considered, centrosome and a centriole, then nucleus. So all these you can find in the eukaryotic cell, so which is basically present where? All of this is present in the cytoplasm itself, right? So in your Selena, I think you can open the page number. I'll just tell you this. About protoplasms, they have mentioned certain features. Okay, so biologists have been using the term protoplasm, okay, for a long time. So they are using this term. So what exactly is this term? Okay, so now by this they mean the living substance in an organism. So what exactly is a protoplasm then? So we'll just highlight the Selena. Okay, what they have given about the from Selena. Okay, so that is for a protoplasm. So protoplasm, according to Selena, or according to in general. Okay, so what exactly is protoplasm? So protoplasm, when we mention, so this is a living substance in an organism. So why do we say that this is a living substance in an organism? So what exactly is happening there? So this is a living substance organism. Okay, then what else? Yes, so thus living substance or protoplasm is contained in this. We can the Selena here. Just a second, I think there is a network issue or not, maybe. Yeah. So in that you can also consider that this living substance or the protoplasm is contained in the cell. Okay. So the protoplasm has been described as a translucent fluid. Okay. Somewhat colorless, grayish or brownish. So we can make a note of this also. So it is what? It is a translucent fluid, right? So it is a translucent fluid. Okay. And somewhat we can mention this to be colorless there, or it can even be grayish. Okay. And then brownish, so this can be considered. So this can be brownish as well. So these are the given points. So a lot of you are having a confusion on what exactly is a protoplasm. So it is nothing but a cell itself along with the cytoplasm and the nucleus. So you can also see it is a living substance mentioned. It is a translucent fluid. So what exactly it is? It is a cytoplasm into which you can see many different organelles that are suspended. Okay, so the chemical composition of protoplasm is very complex. Okay, so it is very little from one cell to the other common elements included in the composition of protoplasm. So there is a carbon, hydrogen, oxygen, nitrogen, sulfur and other them. So these elements are in the form of specific compounds. So you can find a specific compounds there. So what I'm reading, so I'm basically reading from the Selena itself. Okay, so what is mentioned there? So just like every other cell, so we cannot exactly say like what exactly is the composition in this case. Okay, so here you can just make a note, like the composition is going to vary depending on the type of organism and where it lives. So based on that, you can find different composition. So all those in all the cells, so that is iron, then phosphorus, whatever is mentioned, and with that these elements are in the form of specific compounds such as water. Okay, then proteins, so mainly you can find minerals in there. Okay, so there are a lot of minerals you can find. Along with these minerals, so you can also find there are macromolecules like carbohydrates are there, proteins are there. Okay, so then there is a water which is there, fats are there. Okay, so there are fats, and you can also find certain salts, so that is mineral. So all these are found in the protoplasm. Okay, so it is also true to say that it is impossible to make an accurate chemical analysis of protoplasm because it ceases to be protoplasm as soon as it removed from the organism. So in that case you can just, it is the total living substance of a cell, so that is the cytoplasm and then the nucleus. So cytoplasm and the nucleus can be considered as the protoplasm there. So this one we can just write in bracket. So this is nothing but cytoplasm plus nucleus. Okay, so which can be considered as the protoplasm. All right. All right then, so one of the cells, I'll just discuss like protoplasm, the different organelles. So when we mention cytoplasm, so what all are there? So let's have a look at this. So I'll just explain it here, just a second. Yeah, so there is a cell. Hi Astha, good evening. Sorry, good afternoon. Okay, so any doubt that you get, you can ask. So I'll just continue with the cell. Okay, so here the one of the example in order to just highlight what all will be there in the cytoplasm, since we say that the protoplasm is nothing but, so there is a protoplasm. So this protoplasm consists of what? It consists of cytoplasm and then there is a nucleus. So this we call as the protoplasm then. Okay, so now let's have a look at the structural arrangement which will be done in the cell. So I'll just draw the cell now. So we'll draw only the cell membrane. So I'm just drawing an animal cell here. So this is a cell membrane. Okay, and inside this cell membrane you can find a nucleus. So that nucleus is nothing but you can find a nuclear envelope. So there is a outer membrane as such. Okay, so then there is a inner membrane. So this one is now a inner membrane, where the outer membrane and the inner membrane are fused. Okay, so they are fused like this. So you can see a fusion between the outer membrane and the inner membrane, so which is basically called as the nuclear envelope. So this is what, this is a nuclear envelope. So this one is the outer membrane. Okay, so this is outer membrane, all right, and this one is inner membrane. So this is inner membrane. Okay, then both of them are fused and both of them together. So now this is both of them together here, so we can call that as the nuclear envelope. So let me just label this. Yeah, so now both of them together, so we can just call this as the nuclear envelope. Okay, so this is a nuclear envelope. So now what exactly is that gap? So you see this gap, right? So this gap or all these gaps what you see here, so here the nucleoplasm is in continuity with the cytoplasm, because surrounding here is the cytoplasm. Okay, and here is what, inside this you can find a nucleoplasm, right? So there is a nucleoplasm, and at some point so you can also find there is a nucleolus. So there is a nucleolus, so suspended in the nucleoplasm you can find there is the genetic material that is present here as such. So it can be DNA or a RNA. So now this particular nucleoplasm, so this is in continuity with the cytoplasm, so that is through the nuclear pore. So there are exchanges that are going to occur this way. So now this pore what you see, so that pore is basically a nuclear pore. So they are in continuity there. So what kind of continuity? So basically certain exchanges will occur from the cytoplasm to the nucleoplasm. So I'll just erase this, just a second. Okay, so these are nuclear pores now. Okay, now other than that you can also find, so there let's draw the endoplasmic reticulum, just a second. Okay, so now this is now a endoplasmic reticulum that I'll be drawing in continuity from the outer membrane of the nucleus. So you can find this endoplasmic reticulum will be in continuity. Okay, so this is the endoplasmic reticulum where you can find, so let's say only a part of it is attached with the ribosomes there. So here you can find attachment of ribosomes onto the membrane, and this side let's not draw any of the ribosome. So we'll draw only one side so that we can label this side it is a smooth endoplasm, sorry, this side it is a rough endoplasmic reticulum, and this side what you see, so since there are no attachment, so this one is the smooth endoplasmic reticulum. Okay, so that means when you observe it under the microscope, so this side will appear smooth. Okay, whereas this side when you observe, so this appears as rough because there are ribosomes that are attached over here. Okay, and that's the reason this is called as the rough endoplasmic reticulum, and this is called as the smooth endoplasmic reticulum. So here the fat has been modified. So there is a modification of fat that will occur in the SCR. Okay, here the protein modification you can see. So once the protein is synthesized by the ribosome, okay, so that protein is now going to enter. So let me just explain you this. So now ribosomes are there. So ribosome function is what? Ribosome function is to synthesize the protein. So you can see the ribosomes which are attached here also, that is on the outer membrane of the nucleus as well. So you can see lot of ribosomes will get attached here onto the outer membrane of the nucleus as well. And throughout this cytoplasm also you can find again lots of ribosomes that are present. Okay, so ribosomes are present everywhere, that is throughout the cytoplasm. And now this one is the endoplasmic reticulum where the protein modification occurs. Then let's have a look at this. So whatever protein is modified here, so that is basically added with some of the carbohydrates. Then you can see some proteins are added, so we can call as a glycolipid, phospholipid, okay, lipoproteins we call. So basically certain modification is done to the protein and they are going to form a vesicle. So now arising from this you can see there is a Golgi apparatus. So let me just form this. Okay, so now arising from this you can find there is a Golgi body. So Golgi body originated from where? The Golgi body is originated from the endoplasmic reticulum itself. So these are disc-like structures that you can find, which is a Golgi body. Okay, and Golgi body when we mention, so you can have a look at this. So there is this Golgi body and there is a formation of these kind of structures that you can see. So now whatever fat is here which is synthesized or modified here in the smooth endoplasmic reticulum and the protein that is modified and synthesized here in the rough endoplasmic reticulum, so that will take a entry over here and so with certain modification, so it will be packed here in the form of a vesicle there. Okay, so arising from Golgi body itself you can find many of the vesicles. So this one is now a Golgi body. Okay, so there you can find a Golgi body and this structure what you observe here, so this is basically a vesicle. Okay, so we call this as the vesicle there. All right, so now so same way itself, so there is certain proteins which will get modified and you can see there is a formation of vesicles from this. So that is nothing but a lysosome which is coming from the Golgi body itself. So now this lysosome will have many enzymes which can perform the digestion function. Okay, so from where this lysosome is coming, bubbling out, or we can say a part of this Golgi apparatus, there is a packed protein. So protein is getting packed over here, so that is a modified protein by the Golgi apparatus and you can see there is a formation of this lysosome. So lysosome we call this as what? Any idea? So what is that? What exactly is the function of lysosome? Okay, so lysosome is called as the suicide bag. Okay, so what is this called? So this is called as the suicide bag. So that is because there is this lysosome when a particular injured cell is there, or let's say damaged cell, so injured or damaged, or we can say infected cell. Okay, and other than that we can also mention some old organelles are present which are not at all working. So now this lysosome is going to burst and it will release those enzymes and it is going to digest the cellular content. Now arising from the Golgi body itself you can also find there is a formation of tiny bits, so a vacuole formation which occurs. So that is this is a vacuole now. So the vacuole formation can also occur, so that is from the Golgi body itself, and that is why this whole thing, so that is endoplasmic reticulum, the Golgi body, lysosome and the vacuole, all of them are considered under the endomembrane system. So now in cytoplasm only you see all of these things. The nucleus I have anyway drawn, so there is a nuclear pore, so the outer membrane is there, inner membrane is there. So now both of them together, so basically we call this as a nuclear envelope. Okay, so there is a nuclear pore that is present. So nucleoplasm is in continuity, so the exchange of things can occur from the nucleus and to the cytoplasm, so that is through the nuclear pores which are there. So basically there is the exchange that can be seen. Okay, and now this whole thing is an endomembrane system. So we'll draw remaining organelles which are present here in the cytoplasm. So other than all of this, there is a mitochondria. So I'll just erase this part, just a second. So now you see this one, just a second. Okay, yeah. So now, well, let's draw the mitochondria and then I'll show some of the other organelles as well. So let's see the mitochondria now. So here we can just draw mitochondria. So where there is a outer membrane, okay, so you can find the outer membrane which is intact as such. Then the inner membrane is basically folded. Okay, so you can see that the inner membrane is folded randomly, and these foldings which are there, so these are called as cristae. Okay, so these are cristae, and this is the outer membrane. This one is the inner membrane which itself is folded to form a cristae here, and here inside this you can find what? So you can find inside this, so basically there is a matrix. Okay, so there you can find the matrix. So this matrix is the one where you can find DNA. It is having its own DNA and also it is having its own ribosome also. So let me just show you this. Just a second. Yeah, so here you can find it to have its own DNA. Okay, so there it will have its own DNA, and you can also find that it is having its own ribosome as well. So this one is what? This is a mitochondria, and that yellow color what I have labeled, so that is actually the matrix. Okay, so now when we mention about mitochondria, just like chloroplast, so chloroplast is also having its own DNA and a ribosome. Here also this is also having its own ribosome and DNA. So now you know this one, the one that I'm labeling, so it is the matrix that you can find. Alright, so these are the different organelles, and other than that you can find peroxisome. So let's say near the nucleus we can also mention, so let's say this is an animal cell, so this is a plant cell, so we can show the chloroplast. So for to make it plant cell, so you just have to draw the plastids. Okay, so plastids I'll just explain, because since I'm explaining about the protoplasm, so it is the animal cell directly. Okay, so animal cell itself can be considered as the protoplasm because it is a living content of the cell. Hi Prasad, good evening. So this one is the animal cell. Okay, so animal cell does not have a plastids, and since we are mainly talking about the protoplasm, which animal cell can be referred to directly, so now you know these are the different organelles. So then there is one more organelle that you can find, so that is the centrosome, right? So there is a centrosome which is usually located near this nucleus, or we can, instead of drawing here, I'll just draw it a little closer there itself, near the nucleus exactly. Okay, so here you can see this is a the centrosome. So centrosome is there, so which will be having many microtubules. So there can be formation of asters. So this is how they are arranged at right angles to each other. So that is each of these. So now this part is basically the centriole, and you can find there is aster formation around this. So there is a aster rays, and on the whole we can just call that as the aster. Okay, so along with the aster rays and a centriole, so this can be called as the aster. Then other than that, ribosome, we just show it like this, but the ribosome looks different. Okay, so ribosome is having one larger unit and one smaller unit like this. Okay, so this is a larger unit of ribosome. Okay, so this is a large unit of ribosome. Okay, so this one is small unit of ribosome. So this is how these ribosomes are drawn anyway, but they are much more smaller. So just for understanding, I have drawn this ribosome as such. So we have all this in the cytoplasm, which is a living content. And when we mention living content, so this is having different organelles there, and in those different organelles different functioning are occurring. So cell membrane I have shown, endomembrane system I showed, mitochondria, plastids, so ribosomes I showed. Cytoskeleton is nothing but when the cell division occurs, so there is a formation of these. So then there is centrosome or a centriole, and then nucleus. So everything is labeled in this cell. Okay, I hope this is clear. Any doubt? So no doubt. Okay, all right. So let's have a look at some of the questions which are mentioned here. So a protoplast is a cell. So what exactly is this? Anybody? What exactly is a protoplast or a protoplasm? No idea? Okay, I'll just explain it myself. So this is protoplast is a cell. Okay, so there is a protoplasm. Okay, so there where you can find a cytoplasm and there is a nucleus. So here you cannot, like, basically when we mention protoplasm, so it is without the cell wall, so there wouldn't be any cell wall. Okay, then other than that, the controlling center of cell is which one? Nucleus, nucleolus, mitochondria and ribosome. Any idea on this? Which is that controlling center? No idea? Controlling center, or we can also call this as the boss of the cell. Okay, so we call this particular organelle, okay, as the boss of the cell as well. So which is that? So it is option A, nucleus. Okay, so nucleus can be considered as the boss of the cell. So now nucleoplasm, it is in continuous with the cytoplasm, that is of a cell, through the centriole, endoplasmic reticulum, nuclear pore, Golgi apparatus. What is the answer for this? No idea? Again, just a second, this got closed. Okay, so the nucleoplasm is in continuous with the cytoplasm. So you can see there is a nuclear envelope like this. So this is a nuclear envelope having fused the outer membrane and an inner membrane. So we can just draw it like this. Okay, just a second. So in that case, this is a nuclear envelope. The outer membrane and the inner membrane are fused there. Okay. So now this one is the nucleoplasm will be there, and surrounding it is the cytoplasm. So nucleoplasm and cytoplasm, they are in continuity, so that is through the nuclear pore. So this is the answer. Okay, so moving on to next one. The main organelle involved in modification and routing of newly synthesized proteins, that is to their destination, is, which is that? The organelle. Okay, so the organelle involved in modification and routing of newly synthesized protein to their destination is, so chloroplast, mitochondria, lysosome and endoplasmic reticulum. So where exactly modification of proteins or fats occurs. So it is not just about the protein, the modification of protein and fat, so that is done by this particular organelle. Okay, so there is a smooth endoplasmic reticulum, which one modifies the protein, I mean fat, and there is a rough endoplasmic reticulum. Okay, so smooth endoplasmic reticulum for fat modification, the rough endoplasmic reticulum side, so there is, the protein that gets modified here. Okay, so modification of protein and fat is done, so that is through the smooth as well as the rough endoplasmic reticulum. So Golgi body originated from what? Lysosome, endoplasmic reticulum, mitochondria, cell membrane. Any idea from where the Golgi body arises? Just a second, let me just drink some water. No idea on this? So Golgi body is originated from the endoplasmic reticulum itself, because let me explain. So you know that there is an endoplasmic reticulum. Okay, so arising from this endoplasmic reticulum, so these disc-like structures, what are there? So they get separated, and you can see there is a formation of these membrane-like structures, where the endoplasmic reticulum itself is forming this Golgi body, and in turn the Golgi body forms the vesicles, where the packaging is done. So packaging of what? Packaging of modified proteins, packaging of modified fats. So all that can be done here through the Golgi body. So which will be basically packing the proteins and fats. So which is basically enzyme also. So now lysosome is having an enzyme. So that is being packed by the Golgi body by forming the vesicles like this. Okay. So by forming the vesicles, so it goes for packaging. So this is anyway endoplasmic reticulum. Is that clear? Any doubt? Alright, so then we are done with today's session. So whatever doubts you have, you can get in touch with on the Telegram basically. Okay, so all you have to do is on Telegram you can just search for Vani Kari ICSE class 9 and 10 biology, and you can find my channel there. So that is Telegram channel. So we have this offer right now which is going on, where you can get 20% discount on ICSE board plus subscription. Okay, and this started on July 17th and it is till 22nd of July. Okay, so now you can just have a look at this. So this is the current price we have right now. Okay, but with the offer of unlock 20, so we have this offer, so where you will be getting a reduced price, and also the saving, you will be saving this amount on these respective durations. So let's say you are going for 12 months of subscription. So instead of 19,838, so you will be just paying 16,863, and you are saving up to 2,975. Okay, so same way if you are going for 18 months of subscription, so instead of 26,450, so you will be just going for 22,483, and this is the amount that you will be seeing. So you can go for any of these months of subscription depending on your comfort, and you will get 20% discount only if you use a particular code. So you need to use the code VANIYT. So this is my code. So with this code you will be getting 20% discount on the subscription. Alright, so this is basically offer is going on, or otherwise on the regular basis it will be 10% discount. So we have a batch, there is a guardians batch which is a all subject batch for ICSE class 9, and the start date was July 6. So here yes, you have an option to watch the recordings, and there are many more sessions which are yet to go live, so you can take up the live session. And this is mode of delivery here is English. Okay, so same way there is a Avengers batch as well, which is again a all subject batch for ICSE class 9. Okay, and the start date was July 14th. Yes, again here also you have option to watch the recording, but yes, there are many more sessions which you can watch live. And here the mode of delivery is English. Okay, so maths, physics, chemistry and bio will be covered. So in the same batch here also maths, physics, chemistry and bio will be covered. So if you are subscribing for class 9, so you will be getting class 10 to 12 subscription absolutely free. So that is you are going for 12 months on the class 9. So you will be getting class 10, class 11 and class 12 subscription absolutely free along with the class 9. So you can go ahead with the 12 months of subscription there. Okay, and also we have this, so subscribe for ICSE class 9 and you will for the same batch, so you have this option for the other batch also. So basically it is you need to go for 12 months of subscription and you will be getting class 10, 11 and 12 subscription absolutely free then. Okay, so introducing community, so never miss on any update from your favorite educator. So you can get the session notes and practice material there. So you can also share your feedback directly with the educator, so that is via poll. Okay, so check out the link to the educator community below, so that is in the description box. Also we have a Bucks Bounty, so this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. So you can find the form there. So you can click on the link. So if you find anything inappropriate content, so basically you can fill that form and inform. Alright, so you can join my channel, so that is Unacademy, ICSE and ISE. Both also you can join my channel. Basically anyway I said in the very beginning, so that is Vani Kari, ICSE class 9 and 10 biology. You can search on Telegram and join my channel, and this is my Telegram link that I have already given. Okay, so you can subscribe to our channels, and that is Unacademy, ICSE and ISE both. Click on bell icon to get notified about the session. Like and share the video. Okay, so you can share the video with your friends also. So if you find it useful, so you can basically spread it across your friends, and they can also start taking up these free sessions which are available. Okay, and also download the Unacademy Learners app. So this PDF, if you need, I can anyway send it on my Telegram or the community, so you can just ping me there. Alright, so download the Unacademy Learners app, and any doubt that you get, you can just clear your doubts on the live session also there on the platform. Alright then, so see you all in the next session. Bye bye everyone, take care, thank you all.